All right, so we're back on page 2 of our notes, and I mentioned in part 1 that I'm actually going to use 2 colors for my drawings, and you can use 2 colors. However, if you don't have like a pen or a pencil, you can just use a pen, and during the different colored part, you can make one line darker than the other. So again, if you don't have your own 2 different colors, you can just use a pen and then make one part darker and one part lighter. And you'll see what I mean when we start our sketches. So we're going to start off with this part of the notes first, and then we'll go to the drawing side. So we are actually going to start off in the very top box, and the top box, the first part of the cell cycle, okay, and that's what this whole diagram is going to show us, all of the steps of the cell cycle. In the first part of the cell cycle, we start with what we call interphase. Interphase is actually probably one of the longest stages of the cell cycle. So if you take a look on the next box over, okay, the cell grows to its mature size. The genetic material appears as thread-like coils, and we call those thread-like coils chromatin. Okay, those long thread-like coils that we sketched on page 1, that's chromatin. The chromatin is actually replicated. Or doubled. The chromatin condenses down into rod-shaped structures, and those rod-shaped structures are going to be called chromatids. Okay, so that's when the chromatin kind of condenses and coils up into the chromatids that we sketched on the first page. By the end of this phase, each chromatid pairs with its copy, and the copy is known as its sister chromatid. Okay, so that's all in box 1. And the 2 are joined by this central area. Okay, and this central area that connects the 2 chromatids together, if you remember from our front side, the central area is called the centromere. Okay. Oh, we forgot a box up top here. The cell grows to its mature size. And this is called the parent cell. Okay, so we've got the words written down for interphase, and now we're going to sketch the picture of interphase. So if you kind of move over to the picture portion, make sure you've labeled stage 1 as interphase, and the next thing that I want to show you is that this whole circular structure is the cell, okay? And then this inner portion on the inside is actually the nucleus. So it might not be a bad idea for you to label what each part is, just so that you know. And this whole area is the cell, okay? All right, so during interphase, like I said, this actually takes probably the longest of all because the chromatin has to be doubled, and replicating DNA takes a rather long time. So here is the initial strand of chromatin, and remember, chromatin is found in the nucleus. Okay, so that's my initial strand of chromatin. And here's my other color. So if you don't have another color, just make your next line a little bit lighter. So if you look in your notes box, the chromatin is replicated, okay, or doubled. So the reason this process takes so long is because every single piece of information has to be doubled. And so here's my my initial strand of chromatin, and now here's the doubled strand of chromatin. So maybe you put the new strand, that's the coming from the original strand of DNA. And remember, all chromatin is DNA. Okay, so that's our first portion of the cell cycle, which is interphase. So just double check that you have all these parts labeled, as well as the two different colors showing that this was the initial strand of DNA or chromatin, and this is the new strand of chromatin. So after interphase happens, we jump down to the next part of our notes. So we're going to focus on the words first, and then we'll jump over to the drawing. Okay, so this is where mitosis begins. So part 2 of the cell cycle is called prophase. Okay. Now, during prophase, this is where mitosis begins. So I'm actually going to put a star here showing that this is where mitosis begins. Interphase is not a part of mitosis. Interphase is just a whole part of the cell cycle. Mitosis is more specific. So mitosis begins, the centrioles or poles appear. And we've actually talked about centrioles before when we talked about parts of the cell. So the centrioles appear and they begin to move to opposite sides of the cell. Spindle fibers form between the poles and the nucleus disappears. Okay, so in prophase, we get these little spindle fibers that start to appear. And I'm actually going to use another color. Okay, the little spindle fibers kind of look like this. Almost like these little star-like structures. Okay, and at this point, the chromatin has sort of condensed. And we'll sketch that in a second. And then we get these spindle fibers that start to appear. And these are the spindle fibers. You'll notice the nucleus is no longer here. Okay, so the nucleus has disappeared because we need the whole cell now sort of for room to kind of get everything situated and ready to divide the cell. So the last thing I'm just going to add in is that the chromatin has now condensed to form chromatids, which are tightly packed coils of DNA. And remember, they get connected in the center by the centromeres. Okay, so this is just showing that the chromatin has now condensed up into the chromatids, and they've paired together to form a chromosome. Now, in your body, we've actually got 23 pairs of these in your body cells. We're only going to use 2 for the notes because we can't fit more than that in our drawings. The last thing I might just label is that this is the centriole. And the job of the centriole is kind of like an anchor. And I kind of use this analogy. The centrioles actually sort of shoot out these little spindle fibers. And the job of those spindle fibers eventually is going to be to attach to each of these. And eventually they're actually going to get pulled apart. So the centrioles are there, and then these little things, I like to call them spidey webs. They almost look like what Spider-Man sort of shoots out from his little gadgets, are called spindle fibers. So the centrioles shoot out those spindle fibers, and these spindle fibers are eventually going to attach to the chromosomes. Okay, so that is the second stage. And this is where mitosis begins. Okay, part 3 down here. Our next phase is called metaphase. Now, during metaphase, the chromosomes align in the middle of the cell where the spindle fibers attach to their centromeres. So remember, in prophase, they sort of started to condense and everything started to form, and now these little spindle fibers are going to actually shoot out and attach to our chromosomes. The key thing to take away from meta, I always say meta begins with an M, think meta, middle. During metaphase, the chromosomes line up in the middle. So at this point, our centrioles have spread out, our chromosomes are aligned in the middle, and the spindle fibers sort of shoot out and attach to those centromeres. Okay? So that's your sketch for metaphase. We're going to pick up in part 3, finishing up the cell cycle.